see what the hell is. Oh man. Hi, everyone. All right, do you have any questions for me? This might be a very short class then. All right. Well, if you got no questions for me, I have some for you. Why don't we go through the post-test, which I'm sure you can find.
Actually,
one of you has. So we'll do some breakout rooms.
Hey, Dr. Pitt.
And then we'll go through that. And if any questions come up along the way, you can, there's some on the form. Okay, cool.
Yeah, sweet. Thank you.
Good to know. Hold on. Let's see this. I'm supposed to get emails on these. Didn't work. All right. Okay, so let's go through these then one at a time. Do neural crest cells arise from the neural tube? I'm confused on whether they're considered part of the neural tube that breaks off, or are they part of the ectoderm? And then we'll explain neural arches a little bit more. Explain the notochord. Okay. Where is it and what is its function? And explain neural crest again. Okay. The quiz question confused everyone. So I'm just going to accept anything on that one. The idea behind that quiz question was to think about what's making up our peripheral nervous system. So let's start off with our neural crest cells, because that came up twice. And we'll talk about the notochord, and we'll talk about some neural arches. Does that sound all right to everyone? And then we'll hit radial glia. And we'll talk about inhibitory neurons too. I don't know if Y is the best question on that one, but we'll distinguish between our excitatory and our inhibitory neurons. Okay. All right, there we are. I'm gonna get our questions brought up. Okay. So we're going through neurulation. That's kind of the big topic of today. So we're breaking off the nervous system from the rest of the ectoderm. All right, are we familiar with our 3 germ layers? Excellent. Okay, I see some nods. So there's 3 basic germ layers. We have the ectoderm, mesoderm, and endoderm. All right, we don't really care much about the endoderm and mesoderm for today's purposes. They're great. So the endoderm is going to form the gut, some lungs, liver. The mesoderm is going to make muscles. You probably care a lot about that. I think those are great too. It gives neurons something to talk to. to make the skeleton, the inner skin. The ectoderm is the star of today because that's where we make our nervous system from and the rest of our skin. So we're picking up after the time of gastrulation. And so we have our embryonic disc that's going to have ectoderm right here. There's a kind of a swelling in the middle of the mesoderm, and then there's a sheet lining the endoderm. Now there's more to this. It's going to wrap around and help make the yolk sac. Doesn't matter. We got 1, 2, 3 layers. And what we're trying to do here is to make a tube inside of here. All right, we're a series of tubes. One of the tubes that we want to make is the neural tube. And so with neurulation, what we're going to do is start to have cells migrate inward. So we're going to get a bit of an invagination. We're still all ectoderm on the outside. Our mesoderm and endoderm are still below it. The mesoderm is going to make something that we do actually care about for today, and that is the notochord. So the notochord is going to be sitting just below the center of our soon-to-be neural tube. So right there along the midline, the notochord is going to help establish that. So this is a transient structure that's going to hang out just early on in development, and later on, it's just going to form those intervertebral discs. But early in development, it does a lot of really nifty stuff. So initially, it's well attached to the overlying ectoderm. So this midpoint is anchored, and the movement of cells toward the midline causes it to swell up here. Right, so we're going to get this neural groove in here. Now we're going to have fusion of cells right here. So as this continues, we're going to reach the point where we're touching. Now the cells that are destined to be neurons, let's make those purple. It's Friday. These are going to be able to stick to each other. And the uncolored future skin cells are going to be able to stick to each other. But the clear and the purple, they don't mix. So they don't have the appropriate proteins to grab onto one another. So when these purples start grabbing onto each other here, they're going to fuse and break off from the rest of the overlying ectoderm. Now, this isn't going to be perfect. There are going to be some cells that are destined to be neurons, but they don't stick in the neural tube. That's what floats away to form our neural crest cells. And these are going to make anything that lives in a ganglion. So the neural tube is going to go on to form the brain and the spinal cord. So to make upper motor neurons, lower motor neurons, for example. The neural crest cells are going to make our primary sensory neurons. So they're going to make the dorsal root ganglia, for example, and those autonomic ganglia that you find scattered throughout the body. So they're going to form parts of the peripheral nervous system. So when you're thinking neural crest, what you need to think is ganglia. So any place where we have a big collection of neurons, but it doesn't live in the brain or the spinal cord, they must have been derived from the neural crest. These are going to move away and create those peripheral ganglia. Now that doesn't mean that they make the entire peripheral nervous system. They definitely make the sensory neurons that innervate our muscles, joints, and skin, and the post-ganglionic autonomic neurons. But the pre-ganglionic autonomic neurons and the motor neurons, they're going to be derived, of course, from the neural crest. So we still have this notochord here. So there was another question, what does the notochord do? So besides acting as kind of a hinge that defines the center for us to fold around during neurulation, after that, then it serves as a source of guidance molecules. So it's going to release soluble proteins that are going to instruct the nearby neurons to become motor neurons. So it's going to create a diffusion gradient. Really high concentration of stuff here. It's going to get much lower near the top. So it's going to define the more basal portions here, and it's going to say motor. Now the outer plate is going to also release a different set of instructive cues. So we'll have a high concentration up here from those derived at the top. And this is going to create, it's getting a little messy, hold on. This is going to define sensory. And this is what helps establish that division in our spinal cord, for example. Why are the motor neurons all down here in the anterior portion and the sensory relay neurons up here in the posterior portion? Well, it's because early on in development, they were exposed to different developmental cues. So the notochord is going to be a very important determinant of where are you in this future spinal cord. If you're close to the notochord, that means you're at the bottom. You're in the future anterior portion of the spinal cord. So you should be a motor neuron. If you're further away, you get a lower concentration of the become motor neuron cue. You get a higher concentration of the become sensory neuron cue. So an example here would be your bone morphogenic proteins. And over here, sonic hedgehog. It's a different set of guidance cues. They're going to activate different genes, and thus they're going to create different neurons. So the notochord is there to hold on during neurulation. After that, it's going to help establish which neurons are motor neurons. And then after that, it's going to get out of the way. It's going to disappear and become little spongy discs. So hopefully that clears up the notochord as well as the neural crest. So neural crest cells create all the parts of the peripheral nervous system that have their cell body outside of the central nervous system. But the motor elements and the preganglionic autonomic neurons come from, of course, the anterior and lateral horns of the spinal cord. So those are going to be derived from the neural tube. And that's just to remind us that we have a mixture of neurons in our peripheral nervous system. Some are sensory, some are motor. So we have that pair working together, and then we have our pre and postganglionic neurons working together. I don't think it did that effectively. Let's see what else we got here. The neural arches. Okay, so here we're talking about stuff that is kind of peripheral to the nervous system. So these would be the bones. These are important though, right? So our vertebrae are there to help protect the spinal cord from damage. Let's see if I can get us a picture of this. Is that this? Hold on. There we go. All right, I just got to share. Are you guys ready to not have to do this remotely? I think that'll be nice. Okay, maybe you can see this now. So, just at least show it. There's pictures in my way. Hold on. And I have no mouse. Okay. Hopefully Zoom will get out of the way. Cool. All right. So we're going to be building the vertebrae around the spinal cord. And we're going to do that from a few different precursors. So the adult spine, we're going to have our spinal cord in the middle. We'll have some gray matter in the center. And surrounding this, we're going to have our bony structures that are going to be kind of fused together. We're going to have some facet joints holding stuff together. Imagine this sweeping down. I'm sorry, I can't draw in 3 dimensions. And then we're going to have in the back that vertebral arch there, whatever the hell it's called. I don't really care about bones. But these are really nice bones because they're going to hold the spinal cord in a nice, secure location. And this drawing is pretty well crap. But you get the idea. There's a few different bony parts that are going to surround the spinal cord. So you got your body up front. That's going to come from that centrum that they're labeling. You know what? You can't see what I'm drawing. God damn. How dumb am I? All right. I hope this doesn't stop everything. Okay. Okay. So we have bone. All right. I'm darkening that up. Gray matter, not bone. Bone surrounding our spinal cord. So we built this around the spinal cord during development. All right, just like we went from a little sheet of cells, we expanded, and now we have a big old tube of cells that's not just hollow. There is a little canal right in there. So what we did was take a couple of different precursors. So we got 2 different neural arches here that are going to grow together and fuse to form that single bone along the back that you can feel here, and then the central will become your vertebral body. These will grow together too, so we can create a whole vertebrae. So we're going to be fusing together these, the word is escaping me, these more primitive structures. Whew. So as we develop, we're going to be putting things together. So the nervous system is going to grow, the bones are of course going to grow, and some are going to grow together. So the neural arches are going to help enclose the back of the spinal cord, right? So that we're encased in bone. Now this doesn't always work. Just like my screen sharing here. Okay. So sometimes we just have the failure of bones to fuse. Sometimes we have failure for the whole neural tube to fuse. All right, and then there are regions in between, depending on how much of a failure we have. We can have bulging out of just the meninges and neural tissue, but it's still closed. But when we have failure for neurulation, then the whole neural plate is exposed. It's not called a neural tube because we didn't fold it up. And so these are the different degrees of spina bifida. So the neural arches are just those precursors that are going to form the vertebral arch over there in the back. That's all they are. These should grow together and they should fuse so that we encase our spinal cord fully in bone. They don't always do that though. And when that's the only problem that's occurred, likely you're dealing with spina bifida occulta. Apparently it's a fairly common problem that people don't know about because it doesn't really do much. You definitely know whenever you have failure of neurulation to occur. So in that case, nervous tissue is exposed to air and you have severe problems. Okay, then the last question is about crawling on radial glia. So so inhibitory neurons do this, but they do it in a different area. So they're going to crawl on radial glia down there in those ganglionic eminences. Okay. So whenever we're building our cortex, just imagine this is the cortex, right? It's wrinkly. We got these gyri and little indentations, you know, a little sulcus here, sulcus there. Let's not get too caught up in how bad this is. And then there's, you know, thalamus. You have We'll go ahead and put that in there. We're going to have our basal ganglia. We're going to learn about those in here. So we have some nuclei down below, but where the magic happens is here in the cortex, just that outer bark. Now this is filled with both excitatory and inhibitory neurons. And if you were to go ahead and zoom in here, what you'd see are layers of neurons. We have a multiform layer and the 6th layer where you get a variety of stellate and pyramidal neurons. In the 5th layer, we have the large upper motor neurons. Sometimes they're called Bet cells. These are going to project down below. That's where their cell body lives. In the 4th layer, here you're going to have slightly smaller neurons. But what you'll see, and I won't belabor you with drawing all this crap out, there's distinct layers of different types of neurons in our cortex. So it has what we call laminar structure. It's layer upon layer of neurons. And the way that happens is, of course, by crawling along radial glia early in development. So we build it one layer at a time. The first one we built, just like in this drawing, was layer 6. And then we built layer 5, 4, 3, 2, 1. Later on in life, we had the invasion of little inhibitory neurons that are going to form local circuits right here, but don't necessarily fit in nicely to single layers. They're kind of scattered throughout. We'll talk about these later on. I think in lecture 23, something like that. These weren't derived from the local radial glia. So, kind of a mature cortex, if you blur your eyes and then close them. Now, earlier, that cortex was a little bit smoother, right? And we have our, down below there, here's our ganglionic eminences. So we're building the cortex here. Here's where our excitatory neurons come from, future cortex. But the inhibitory neurons are derived a little more ventrally. Ganglionic eminences down here, and what they will do, there's radial glia here. They're going to crawl along them for a little bit, but they actually have a much longer journey to go up and get into the cortex. So they're going to crawl long distances to make these inhibitory neurons here. Why? I don't know. My guess is that the radial glia in the cortex just don't know how to tell the newborn neurons to be an inhibitory neuron. So the transcription factors that the mother cell hands down to the daughter cell just turns them into excitatory neurons. So that's all they make. And you have a different set of radial glia down here, so they make a different set of neurons. Now, why don't we intermix them up here? No idea. That's just the way it is. I know that's not a good answer, but there's usually not a good answer to why in biology. Somehow it increased the reproductive fitness of the organism. I guess that's why. But it is true that we have two distinct populations. We have our cortical radial glia that are going to make excitatory neurons, and then we have more ventral radial glia in the ganglionic eminences that make inhibitory neurons. Not surprisingly, those neurons away from the cortex, this is coming attractions here. So down here in the basal nuclei away from the cortex, so very deep, that's why it's called basal. And I hate it, but we call it the basal ganglia. These are definitely within the central nervous system, but they're called basal ganglia. Makes no sense. Anyway, these are largely inhibitory neurons. In fact, they're almost exclusively inhibitory neurons. So later on in lecture, whatever the hell it is, when we go through the basal ganglia, it's going to be a bunch of inhibition because most of them are derived from those ganglionic eminences because they're not in the cortex. They are subcortical. That's why it's called basal. It's called ganglia because someone fucked up. They should be called the basal nuclei, and sometimes they are. But most of the time they're called basal ganglia. All right, I'll write some answers to those questions and make sure they get up on the site. Sorry, I didn't get emails on those. All righty, So I think that addresses those. Are there any more questions? Okay, well, if they come up, type them in here and I'll get to them. Now, why don't we break out into some smaller groups and go through the post-test. So chat with your classmates, see if you can come up with the right answers, and then we'll run through those and talk about why the correct answers are correct, and if any questions are bad, why they're bad, and I'll just forget about them. Any objections? Any enthusiasms? Okay. Let's see, 6 to 7 per room. I don't know about all that. It seems like too much. Okay, that sounds better. All right, I'll see you in a bit. They are now open. Yes. All right. Hopefully you got all your answers in by 11:41. Anything that you wanna go through? Is anything kind of tricky?
Can we go through the first question?
Or did any questions come up? Okay. We will go through all the questions, and we'll start with the first one. I think that's a great place to start. We'll end on the last one. All righty. Oh, I need to share. Actually, you know what? I don't need to. You can see these questions, I'm sure. Okay, so the first question says the notochord aids in nervous system development by. What did you think on that one?
All of the above.
Does it sound like it attaches to the neural plate? Is that something that we encountered in the notes or the words I said right before this? Okay, I have a slight nod. Good, okay. So maybe the first one. Does it induce neural cell fates in the ectoderm? Hopefully some nods there. Does it secrete proteins that are going to promote motor neuron development? I hope so. So it sounds like it's doing all those things. So the notochord is pretty important. It's going to be an anchor and it's going to be a guide. I think those are some words that I've used in the past. It's an anchor because it attaches to the neural plate and gives it a place to fold, not that violently. And then it's going to act as a guide. It's going to say become nervous system, and then a little later, more particularly, become motor neuron. So the notochord is pretty important, but it's really only important in development, and then we get rid of it. The second question asked you if the nervous system was derived from different layers, and you all picked ectoderm, so that's great. You get that the notochord is from mesoderm. Telencephalon forms the cerebrum. Excellent. Radial glia are going to act as tracks. No one picked that symmetric cell division. I'm glad to see that. They don't differentiate into neurons. Their daughter cells do. That's cool. Let's see. The bundle of nerve fibers shown here. Okay, so we got a picture. Hopefully you're looking at the same thing that I am. We're looking at, in this case, it's going to be a sagittal plane there, so we're cutting through. We can see the vertebral bodies there on the left, arches on the right, spinal cord running down the middle. There's a big red box around a bundle of nerve fibers. I've specified that it's a bundle of nerve fibers. So could it be the cauda equina? Absolutely. The phylum terminale, that's probably in there, but it's not a nerve fiber, so I wouldn't pick it. The lumbar cistern is there. It's not a bundle of nerve fibers, though. But lumbar cistern's a bunch of CSF. So I wouldn't use any of those terms. I would use the term called equina for the nerve fibers that are shown there. So that's a collection of the ventral and dorsal roots that are running down. They haven't fused yet, not till they exit. So the nerve fibers of the cauda equina. They are in the lumbar cistern. This is true, and there is a filum terminale connecting the spinal cord down there with the bottom of the spine. Okay, this was a tricky one. The next one, turns out it was tricky. Which of the following best illustrates the overlap between dermatomes? So can someone please remind me what a dermatome is so I know what we're talking about?
Area that a single spinal nerve gives sensation to.
Hannah Brown, where you at?
It's like where
Anywhere?
a spinal nerve, like it's where its path goes, where it innervates.
Let me see if I can find you in here. Don't make me hunt you down. You're here. Hey, there you are. Found you.
It's like the
Thank you for
path
volunteering. Tell me about it.
of
Tell me about
innervation.
dermatomes.
Okay. It's like where a spinal nerve like innervates. It's like the path.
Okay, you probably told me that earlier. I just realized that my speakers were muted.
Yeah.
It's not embarrassing at all. Okay. You're taking classes from this guy, by the way. Okay. So the band of skin that we're innervating. So hopefully we're dealing with something to deal with sensory function. So compression of a nerve root produces both sensory and motor deficits. Probably not. Probably doesn't have anything to do with the overlap between dermatomes. How about that second option? If we were to sever a single nerve root, we have no noticeable loss of tactile sensation. This is pretty well true. So just a single root doesn't do much. You'll have a little area, perhaps, but you won't really notice it. That is definitely going to show us the overlap there. You might be tempted to pick the shingles there, but the fact that you get a unilateral band just tells you have one side of the spinal cord that's infected. It has nothing to do with the overlap. You're probably tempted to pick all of the above. I see 11 of you were, because it says all of the above there. I think I mentioned I know this trick, so don't fall for that. Okay, so which neurons are formed by the neural crest? And this was one of those asshole questions where more than one answer could be correct and you got to select them all, but 44 of you did great here with that. Postganglionic autonomic neurons for sure. Somatosensory neurons. That neural crest question at the beginning was timely, that's for sure. We all got our notochord in the cartoon, so that's cool. And then we had some pretty picture to look at the end. And most of us have picked that it's radial glia. So why isn't it excitatory neurons? What I'd like you to notice is how these fibers are running. So if you look in panel B, that's showing you that little box in panel A. All right, we're staining top and bottom. And then we have these fibers that are kind of running all the way from the ventricular to the apical surface. That should be dead giveaway that it's radial glia. Now these will give rise to excitatory neurons, but the excitatory neurons aren't going to span from top to bottom. They're going to fit into a smaller layer. The radial glia are going to go from top to bottom. And these are indeed radial glial fibers. So that's what they look like in real life, in case you're curious. I did have you muted, and now I see those. Okay, all right. So that explains the complete lack of sound so far. All right, well, I feel better about that. Are there any questions before we get out of here, have a weekend, and then not meet over Zoom? Hopefully for the rest of the semester. Because this is great, except when it's not.
I do have a quick question on
I
that.
can hear you. Great.
Yeah, fantastic. On that last picture, there's radial glia, and then where the green, the whatever was stained green, was that excitatory neurons?
Yeah, yeah. So what you're seeing down there in C, you can see neurons there, C and D, and then you can see them a little bit in E as well. B, definitely just looking at radial glia. Now, what I want you to keep in mind is that you see a whole shitload of green down below. So look in figure A there. Where are you seeing most of the green in panel A?
Far, way further down on the picture.
Exactly. So those would be our ganglionic eminences. So most likely what we're looking at here would be the migratory inhibitory neurons. So they're kind of working their way into the developing cortex here. But as far as panel B and the fibers that we see in E, we should be thinking radial glia because we're going from bottom to top. If it were excitatory neurons, we wouldn't light up the ganglionic eminences like we're seeing below. It'd be just kind of restricted to the more dorsal portion of that panel. But you can clearly see down there the ventral ventricular zone. That's lighting up ganglionic eminences, probably the inhibitory neurons that show up. Does this really matter? I don't know. Probably not. It's a nifty part of development that our excitatory neurons stay local and the inhibitory neurons are the ones that have to migrate. I don't know why, but that's how it is. And now you know.
Appreciate it. Thank you.
And these recordings will be posted somewhere, and that where is the class website. I'm actually recording these. It's not that I remember, it just does it automatically. So they'll be up there in case you want to look at them. And hopefully we don't have to resort to this again.
So for this like post-test thing, when you hit submit, it just says like thank you for your submission. It doesn't tell you like which ones you got right and wrong. Is there a way you could turn that on so that when we use them to study for the exam, we can know
Yes.
what we need to review?
So first time around, I'm going to leave it like it is. I forgot to do that on the first one. So this way, you know, when we're in class going through these, there's still a little bit of mystery, so you can actually, you know, have some discussion after the fact and say why you chose what you did. And you probably wouldn't be inclined to do that if you knew that, like on that last question, if you didn't know that it was not excitatory neurons, you probably wouldn't be inclined to tell me your rationale for that. So I want to keep it a bit of a mystery. But yeah, so after they get submitted and we talk about them, then I'm going to switch it back over. So as you go through the post-tests, it'll tell you what you got so you can use these to study.
Thank you.
You're welcome. All right, well, we'll call that a class then. And I'll see you next week. We'll be going through lectures 3 and 4. We have nothing on Monday. What are we talking about? I'm pretty sure we're going through action potentials and probably resting membrane potential, ion channel, stuff like that. So this is good stuff. I'm excited. And I hope you are too. If not, you will be. That's the whole purpose of creating ion currents, to excite us. If you have any questions, let me know between now and then. Have a great weekend, a great 3-day weekend, that is. I'll see you next week.